LECO ChromaTOF's built-in spectral analysis tool software package enhances your ability to smartly interrogate high-resolution mass spectral data to locate chemically related analytes of interest. In this example, we are trying to locate sulfur-containing species in a two-dimensional GC separation of a feature-rich sample of heavy fuel oil, since these sulfur compounds are regulated to very low levels worldwide in marine fuel oils. GC by GC provides significant separation power, but the high level of hydrocarbons and general complexity of the sample can make it more difficult to zero in on just the lower level sulfur-containing compounds. To generate spectral plots that are easier to interpret, we will be using the concept of mass defect, the amount of difference each atomic species has from its nominal value. Since every atom has a unique mass defect, predictable differences in mass defect can relate back to series of mass formula, and then to chemically related structures. All of the mass signals throughout the separation for this heavy crude oil sample are combined into one summed region spectrum, which we can see here in the spectral plot. When we bring up the mass defect view, we can now see each mass signal plotted with its corresponding nominal mass on the x-axis and the mass defect on the y-axis. With spectral analysis tools, we can write mass defect series to search for masses that follow a specific pattern. In this general sulfur series, mass formula that contain a single sulfur atom, a sulfur oxide group, or a sulfur dioxide group will be highlighted on the mass defect plot in various shades of yellow, with formula that iterate by a CH2 group, extended for all RDBE values. When we apply this mass defect series to our plot, we're immediately able to see the colorization of masses belonging to these sulfur-containing formula on the plot. Scaling the mass defect plot helps to arrange the pattern of the dots in a way that allows us to more easily see groupings. With petroleum samples, the Kendrick scaling is commonly used, which scales masses by iterative groups of CH2. After applying a filter to hide all unclassified masses, only these sulfur-containing species remain, so we see only masses that correspond to signals of interest. Next, customizing bubble size can help us see the distribution of our mass signals, with larger bubbles corresponding to higher abundance masses in the sample. Once we've identified some masses that may be of interest, we can select and plot them back onto our GC by GC plot and check for corresponding peak identifications. For example, here we select one of the larger yellow dots from the sulfur-containing mass defect series and show it on the contour plot. Immediately, we are able to pull out at least one section of the contour plot that corresponds to this mass signal. When we click on one of the peaks, we are able to confirm its identification as dibenzothiophene using the deconvoluted peak true mass spectrum that has automatically been matched against the NIST library. If we select the next mass from the mass defect plot in the same mass defect series row and plot that, we can see the next grouping of peaks that correspond to dibenzothiophene structures with an additional CH2 group, the C1 or methyl dibenzothiophenes. As we select additional masses and add them to the contour plot, the clear elution pattern of these C1, C2, and even C3 dibenzothiophene emerges. The ratios of these methylated dibenzothiophene are important geochemical thermal maturity markers that we are able to clearly and easily highlight using the scaled mass defect series. With spectral analysis tools, finding these sulfur-containing species in heavy fuel oil is simplified, demonstrating a workflow that is beneficial not only in the petroleum sphere, but also for environmental or non-target discovery applications to pinpoint low-level analytes of interest in high mass resolution analyses of complex samples.